Hello everyone, this is the first screencast for Biology 241, Christina Howard's lecture. And this screencast covers, we'll see how far we get in chapter 4, which is the tissue level of organization. So, the definition of a tissue is groups of cells performing similar common functions. And we'll talk about more details on those functions as we move along, but for now, just know that these are, things like secretion or protection, etc. So the tissues of your body are those which give your body its both structure. So you've got layers of different kinds of tissue, muscle, fat, etc., but also its function because the cells of your tissues perform the metabolic functions of your body. So let's get started. So the tissue types, you've got 4 overarching ones. I'm going to switch colors here because I don't like this red. It looks angry. Let's do purple. So these are the 4 major tissue types right here, and they are epithelium, connective tissue, muscle, and nervous tissue. And in order to sort of set the stage for the rest of the lecture, I'm going to start like I did in lecture by telling you a story. And that story is where tissues come from, and also the story of how you and I came to be here. So let's start. Oopsie, I want to go back. So, a long time ago, you were an ovum and a sperm. And eventually, those 2 cells would meet at the moment of fertilization. And after that, we have the first cell of a new individual. So the first cell of a new individual is called the zygote. I want to be clear about that because people often say zygote and fetus and embryo and use those terms incorrectly relative to the stage of a developing person. So in an effort to combat that, I'm going to be very intentional with my language here. So once the first sperm gains access to the egg, all others are turned away, and then you have a zygote. So, I'm going to make my dot a little smaller too. Okay, so here we have a human egg cell, and these are paused in metaphase 2. If the egg gets fertilized, now we call this thing the zygote. Hooray. Then it will complete meiosis, forming the second polar body. So we go from 1 polar body to 2 polar bodies. After that, meiosis is over, and regular old mitosis gets underway. So you aren't a single cell for very long, it's just how you start. So almost immediately, the new cell that is you, the DNA from mom and dad, decondenses and combines, and that's your new genome. Congratulations on your new genome. And then it gets to work dividing. And we call this division cleavage, specifically because in the initial phase of cell division for human development, you're getting an increase in cell number, but a decrease in cell size. And that's because the cell division is constrained by this membrane, which is called the zona pellucida. So the cells are actively dividing, but they're trapped within a connective tissue sphere, basically, and so they don't have any room to get bigger. So more cells, smaller cells. Okay, so it's always important to talk about location. So in every term we teach you this class, I'm going to ask you to know the definition of the term, but also a location and function. That's kind of the central theme of the class. So it only makes sense to describe the location where all this is taking place. So in the normal course of human development, fertilization happens in the fallopian tube, not in the uterus, as many think. And then cleavage is occurring as the cell sort of gently bounces down the fallopian tube towards the uterus. And this is the time during which you're getting a lot of cleavage, so more cells, but smaller cells. By the time you're about 16 cells, you are called a morula. So an early morula is about 16, and then the increase in size causes you to become a late morula. Once the inside of the mass of cells hollows out and becomes a ball with a little inner cell mass here, then we call it a blastocyst. So by about day 6, that's occurring, and then days 7 through 10, the blastocyst is basically burrowing down into the uterine wall and continuing to change. All right, so here's sort of a zoomed in view of that. You can see the cute little blastocyst sort of docking at the surface of the endometrium. And those uterine glands are going to have all kinds of nourishing substances in them. The placenta is not yet formed yet, so there isn't a shared blood supply, but the glands produce a substance that is really rich in glycogen, so it gives these cells a lot of division fuel, basically. So without going into too much detail about the specifics of human development here, because we'll do that in 242, this little area right here is of particular importance because now, instead of being sort of a uniform mass of cells, the embryo now has 3 distinct layers. So first you were one cell, then you were a ball of cells, and then you were a sandwich. So that sandwich, we've zoomed in on a little bit here. You can see here is one layer, here's another layer, and here's another layer. So 3 different layers of cells. All right, so here's 2 views of the same thing. So now we call this an embryo. I know it doesn't look like what you would think an embryo would look like. It doesn't look like people yet. It's got to fold some more. But the process of the migration of superficial cells into the interior creates the mesoderm. So as you can see here, we've got the primitive streak. And then notice these cells are dividing here, and as they divide, they're moving towards the primitive streak, and then some of them are being chucked off at the primitive streak into the middle, and that forms the mesoderm. So we've got the ectoderm, which means outside, mesoderm, middle, endoderm, inside. So these are called the primary germ layers, and it's because it's well understood that all of the tissues of your body can ultimately be traced back to one of these 3 layers. So let's have a look at that. Oops. I'm going to erase this. And let's pause here. So I'll show you a little bit more about the 3 germ layers in a little bit, but you'll notice that once the embryo has buried itself in the wall of the placenta, it actually starts to do some folding, and that's how it eventually attains its appearance as a more human-looking thingy. So that starts with the head fold, and the head fold is what it sounds like. So the anterior of the embryo begins to fold down and inward, and we get an eye spot, and then that's the first discrete organ that you can see. So as we move on, that process continues. So we get an embryonic head fold, and you even get an embryonic gut. So the gastrulation process where those cells are migrating eventually forms a tube, and that is how your gut is formed. We are, I'm using we meaning humans, we are deuterostomes. So there are 2 possible ways in embryonic development for vertebrates to form, and one is mouth first, and one is anus first, or mouth second. So protostomes are organisms that form their mouth first. Deuterostomes form their anus first and their mouth second, and that is us. So came into the world butt first. Okay, so now we can see that this is starting to look more like a familiar embryo. It doesn't have any limb buds yet, but it's got some other cool stuff, so you can see that it's starting to look more like a person. Okay, so let's go to the next page, shall we? Et voila! Now we have a tiny, tiny human with all the tiny, tiny parts, and you have all of your organs, at least the shape of them, by week 8. So after this point, those cells from the primary germ layers are beginning to move around the developing fetus, and it's going to start specializing into the tissues that we know. So, all right, so here are the embryonic origins. We have the sandwich again, so ectoderm, mesoderm, and endoderm. So the ectoderm gives rise to certain kinds of epithelium, including integument, and the nervous system, all nervous tissue, including your brain, but also your peripheral nerves. The mesoderm gives rise to epithelium as well, specifically the lining and covering epithelium of your body cavities, but also connective tissue and muscle. And the endoderm is epithelium only. so all the other kinds of epithelium. So as you look at epithelium, you may notice like, oh, hey, the epithelia are pretty diverse. That's because each germ layer gives rise to some kind of it, which is interesting. All right, so all adult tissues are derived from one of the 3 embryonic tissues. So let's start by talking about an epithelium. And for this, there are 2 types. So lining and covering and glandular. So covering is skin. Lining is lumens. And I don't mean the measure of how bright a light is. The lumen is the empty void inside of a hollow organ. So this is, let's say this is the cross-section of a stomach, for example. Your stomach's a hollow bag. So this is muscle, and then we've got epithelium. And the epithelium faces an empty space called the lumen. So when you hear me use that word, that's what I mean. Glandular epithelium is specifically designed to produce secretions, so things like gastric juice, saliva, what else? Mucus, those kinds of things. So we'll talk more about those later as well. Okay, so characteristics of epithelia. They're very cellular. And I want to be clear about one thing with regard to cellularity. This is kind of a relative term, so you wouldn't say something is simply cellular. It's really that they're cellular in comparison to other tissues. So think of this as being lots of cells all touching each other. So you're not going to look at epithelium and see like 2 columnar cells and then a space and then another columnar cell and then a space, like a mouth with missing teeth. That's not how it works. They're all touching each other, and that is to serve a purpose. So the lining and covering function is a way to keep your body compartments discrete from each other. They also exhibit cell polarity. And this just means that there is a side that faces the lumen. So up here, the lumen is here. And we call that side the apical surface. That looks like lumer, not lumen. There we go. And the basal surface is whatever the cells are sticking to. So the basement membrane is the extruded membrane and cell junctions that allow the cells of the epithelium to stick to whatever's underneath it. Usually what's underneath it is connective tissue. Not always, but almost always. Another feature of epithelia is that they have a really high regeneration rate. And this is good because of what they're there for, right? So if I fall off my bicycle and scrape my knee, it is not the case that I'm like, well, bye forever, knee skin. It was nice having you. That's not what happens. My body will kick into gear. I'll have an initial inflammatory response. And then those cells get to work doing mitosis, and eventually I just got some brand new knee skin. So epithelia are really good for that purpose. There's a trade-off, though, and that is that because they have higher rates of regeneration, epithelia are more prone to cancer than other kinds of more slow-growing tissues. So can't win them all. Another thing that's a characteristic of epithelia is that they are avascular. So you would not expect to see, for example, we'll come here. So in histology, when you see a blood vessel, it usually looks like a hole. Actually, let's make it pink, because that is more reflective of what you see when you look in the microscope. So if this were a blood vessel, it would be a hole lined by pink. And here's some nuclei because it's lined with simple squamous endothelium. And then you would see some blood vessels in there. So this is super normal to have in connective tissue, not so in epithelia. So no blood vessels. Exclamation point. Let's make that no a little bit clearer. No blood vessels. So you only see these in connective tissue, not in epithelia. Epithelia rely on diffusion from the cells below them to get that done. Okay. So, some general functions for epithelia. One is protection. So, I mentioned the knee scraping example, that's protection against abrasion, but also dehydration. So, there are glycolipids between your dead skin cells that help you not lose water or gain water, and also protect you against chemical insult, because the cells on the surface of you, they're dead. They don't care if they hit a nasty chemical as much as living cells underneath. Another function is secretion. So they make mucus, which is, you guys know what mucus is. It is what it sounds like. And also serous fluid. And you might not be as familiar with this. Serous fluid is just any thin, watery fluid. Usually, although not always, usually contains enzymes. Another important function of epithelia is absorption. So, cells of epithelia that are absorptive control their permeability via membrane proteins. So, let's give you an example. So, i.e., glucose transporters in gut. So, how does glucose get from the lumen of your intestine into your blood? Glucose transporters. That's how. So, the simple columnar epithelial cells, of your small intestine are specialized to absorb nutrients. If you didn't have them, you couldn't absorb nutrients. Bad news for you. Okay, so some general stuff about epithelia. They rely on intercellular junctions to stick to each other. So those cells of the highly cellular membrane stick to each other. They have an attachment to a basal lamina, so that's how cells stick to stuff. And they are involved in maintenance and repair. So this is high mitotic rate. So intercellular junctions, inter means between, cellular means cells. So I'm just going to try and even the most mundane vocabulary, I'm going to try and break down for everybody, because if you get used to doing that, if you get in the habit of it, sussing out new words is easier because you can just look at their parts. So inter means between, as opposed to intra, which means within. Things to watch out for. Okay, so most cells and tissues are adhered to each other and/or, I'm going to add an and here, the extracellular matrix via cell adhesion molecules, which are things like proteins that stick to other proteins, so BAM, and substances that they excrete called hyaluronin, which is an intercellular cement. So you've got a really robust two-piece system. One is an actual large protein molecule or some such, and the others are some kind of ooze that the cell excretes. So the three functions of intercellular junctions include fluid-tight seals. So this helps you keep your body water where it's supposed to be. So blood stays in the blood tubes, intracellular fluid stays inside of cells, extracellular fluid stays where it's supposed to go. It also helps to anchor cells together and act as channels in the case of gap junctions, which I'll show you momentarily. Okay, so it's pretty unusual that cells only have one kind of junction. So depending on how they're arranged, they may be joined together using more complex junction arrangements. So let's look at these. We're looking at the example of an epithelial cell. These happen to be columnar cells with microvilli, which suggests that they are in the intestine. That's not a conclusion I would expect you to be able to draw yet. I'm just providing you with additional information. So if you were like, how did she do that? Don't panic. I did it because I've been doing this a long time, not because I think you should know that right away. Although you will by the end of this. You'll be able to look at a cell and say, it meets these criteria, it's probably one of these. Okay, so tight junctions are the first kind, and these sort of quilt the two membranes of the cells together, so that if, let's say I'm a bacterium, and let's just to, honor tradition, let's make our bacterium green. So there's my bacterium. He's somewhere between a coccus and a bacillus. And let's make him, I'm going to anthropomorphize him a little bit. So he's mean. Look at his frown. He wants to go in here, and he wants to not go through the cell, because if he goes through the cell, he's probably going to get captured and stuck in a lysosome. So he's going to try and go between the cells, but due to the presence of tight junctions, denied. It also creates a fluid tight seal. So let's say I drink a bunch of water. The water is going to have to be absorbed in my stomach and small intestine through the cells, not between them. Okay, we've also got spot desmosomes, so I'm going to go back to my purplish pink color. And these are protein plaques with intermediate filaments. So remember the intermediate filaments from the cytoskeleton from 160. And then those provide an anchoring surface for cell adhesion molecules, which stick into the intercellular space. We also have proteoglycans in here, which is a sugar carbohydrate composite, and these are molecules that are good at attracting water, forming a gel. Hemidesmosomes are just a half a desmosome, and the reason it's only half is because this is where a cell is sticking to something that is not a cell. So in this case, the basement membrane, also made out of proteoglycans. The final kind is gap junctions, and these are basically tiny little tubes made of a protein called connexin. And these form a little array that looks like a flower, but also a tube, and this allows ions to go through. All right, so attachment to the basement membrane is important. So this attaches epithelium to underlying connective tissue, and it is secreted by the epithelium. So these cells are going to sort of dump extracellular stuff onto the basement membrane, and that's going to form a barrier. So these are composed of proteins and glycoproteins. So glycoproteins are protein sugar hybrids. These are actually part of a cell's glycocalyx. Woo! So recall that the glycocalyx of a cell is the assortment of proteins, glycoproteins, and glycolipids that hang off of its surface, and those serve a variety of functions, including but not limited to sticking cells to stuff and cell-cell communication. So we'll keep coming back to glycocalyx stuff as we move through the next couple terms. So I know this is something that you learned about in 160, or at least you should have. You don't get to let go of that piece of knowledge yet. It comes back. Okay, so maintenance and repair. I mentioned that epithelia have high rates of regeneration, and that's true. And that consists of a system where stem cells give rise to two different kinds of cells. So I'm going to go back to, oh, actually, let's look at this aubergine color. Aubergine is a fancy person way to say eggplant. All right, so let's say I have a stem cell. Oops, got to draw. Okay, so this is my stem cell. So when the stem cell divides, it's going to produce two daughter cells, right? So you can't have mitosis that results in 3 cells. It's always ever only 2. So one of those cells, let's say this is a stem cell that's part of the epidermis. One of those cells is going to become a keratinocyte, which is going to multiply and die as it moves upward to form your epidermis and the keratin surface of it. But the other daughter cell is not also going to be a keratinocyte. The other daughter cell is going to be another stem cell. So in this way, there is a constant, steady supply of both keratinocytes and new stem cells. So you don't run out of either. Clever, huh? All right, so covering and lining epithelium, this lines and covers surfaces. So your skin, an outer surface, your GI tract, an inner surface, and your respiratory tract, also an inner surface. So this is a binomial nomenclature system, meaning that there are 2 names, and you need to give me both. So cell shape and number of layers. So when you give me a name of a tissue and it's an epithelial tissue in lab, you need to say something like simple squamous epithelium or stratified squamous epithelium or simple cuboidal epithelium. You need to give me all 3 words because that is the most complete and most accurate description of that tissue. So don't just say squamous, don't just say simple. Please don't say simple stratified, can't be both. Make sure that you're mindful of your terminology. Okay, glandular epithelium. These usually are multicellular glands, but not always. There is an exception. And these are specialized to produce secretions. So secretions come in 2 flavors. Endocrine, these go into the blood, and they stay in the body, hence endo. And exocrine, these go onto a surface. And they can even be cast off outside the body. So spit, for example, earwax, sebum, tears, all of these are exocrine secretions. We categorize these by mode of secretion. So how does the cell secrete what it needs to? We'll look at that next. Type of secretion, so is it sticky, is it thin and watery, what's in it, and the structure of the gland, which we'll focus less on. So mode of secretion means how does the cell accomplish secretion? And there's more than one way for a cell to do this. So merocrine, and I'm going to make an addendum here. You may see these referred to as ecrine as well. Either is fine. Merocrine is the more modern accepted term. But I try to warn you about terms that you may encounter that are synonymous, because I know you guys use the internet to study. So merocrine glands are glands that secrete via exocytosis and without significant loss of cytoplasm. So the Golgi apparatus is constantly producing secretory vesicles, and those vesicles are going to fuse with the membrane of the cell, and then what was once outside, or inside, excuse me, is now outside. Apocrine glands, the whole top of the cell breaks off. So you can see there's this pinching that's going on. So there's a ring of cytoskeletal elements in here that is polymerizing and pinching this off. The nucleus and the rest of the organelles stay there, but the top bit breaks off. So you get phospholipids and whatever's in this little package of phospholipid, and those things break down. So an example of an apocrine secretion is breast milk, but also sweat from apocrine sweat glands in the armpit and groin. In holocrine glands, the entire cell is sloughed off. So we have a stem cell here. It's going to divide and give rise to one stem cell and one holocrine cell. The cells are going to busily fill up with secretion and increase in size. And then when they get to the top, they're going to burst, and the secretion is going to be cell bits and whatever secretion they were making. So in the case of a sebaceous gland, they make sebum, which is oil. So if you have oily skin, what it means is your sebaceous glands are doing a great job. Okay, so we also categorize these by type of secretion. So type, in this case, I'm being intentional here about distinguishing type from mode, because I want you to understand that if I ask you for mode of secretion on, say, a test, what I mean is the previous slide, not this slide. So type of secretion refers to what is the secretion like physically. So serous glands produce watery solutions that contain enzymes. So for example, saliva, tears. Mucus glands produce a protein called mucin, and mucin attracts water in a hydration cell to form a sticky mucus. And there are mixed exocrine glands as well, so these produce a mixture of mucus and serous secretions. So a good example of these is your submandibular salivary gland. All right, so in this case, I promised I'd be transparent with you about what is and isn't going to be on tests, and I will stick to my word here. So although there are structural classifications of glands that you can kind of divide into 3 types, it's not really important that you know this from a functional perspective. So the shape doesn't contribute enough to the function of the gland to make us feel like that's really important, a piece of information for you to hang on to. So don't study it. The next slide is only for example. Hey, look at how diverse these are. But at the end of the day, they're all just an invagination of an epithelium. So, oops. So you can see this is just an epithelium turning inward to form a gland. This is just a fancier version of the same thing, and so on and so forth. So it's just increasingly complex versions of the same little infolding of epithelium. All right, and I mentioned earlier that there was an exception to the multicellular gland rule, and that is goblet cells. So they're called this because in the epithelium they sit in, let me see if I can draw a little bitty epithelium. Very, very lazily. So these are cilia. So this is a ciliated columnar epithelium. And then every now and then in this epithelium, you're just going to see a big white spot. So these are called goblet cells because they have a big blob of mucus at the top, and then the neck of the cell is really narrow, which means that if you were to like get this out of the epithelium, it would look like this, which looks like a drinking goblet. So that's why they're called that. So these are unicellular glands that secrete mucus, and they're located in the lining of the respiratory and digestive tracts. So they're there to do things like trap particulate matter, to prevent it from getting into your lungs, and trap bacteria and lubricate the passage of substances, all kinds of stuff. Multicellular glands are a little bit more diverse, so these vary in size from sweat glands, which are very small, to your pancreas, which is an entire organ's worth of gland. So lots of diversity in glands. Alrighty, so I'm going to stop here video-wise, and when we pick back up, we will talk about connective tissue. That's going to be in part 2 of this video, but right now I have some other stuff to go do, so I will see you guys for the next video. Thanks for your attention.